Hi guys, this is Miss C, and we're going to do our first Kingdom Animalia lesson. It's going to be over early embryology. Early embryology is really talking about how animals develop and where their tissue layers come from and what it means to be more complex and more simple. So starting out with a sperm and an egg cells, which are both haploid, meaning they have half of the chromosomes that a diploid organism would have, you get a zygote through fertilization. Fertilization, of course, is when the sperm and egg come together and create the diploid cell. Now that we're diploid, we're still only one cell, and we know that as humans and all other animals are multicellular individuals, so we need to become more than one cell. And the way that starts out as a zygote is a process called cleavage. Cleavage is when you split this one cell into two through mitotic division. Remember how meiosis and mitosis are different. Meiosis is what created that egg and sperm cell and created haploid cells, while mitosis is just simple mitotic division, keeping this number of chromosomes the same. Imagine taking a ball of Play-Doh, rolling it up in your hands and holding it in your hand. If you were to take a string and drive it through the middle of the Play-Doh ball, you'd create two halves. You can realize that the volume of the Play-Doh, the amount of the Play-Doh in your hand has not changed, but the number of segments you have has. So that would be cleavage, splitting that one zygote into two cells, not changing the volume. This continues on to create cells that are 2, 4, 8, 16, 32, and so on. You can see here that that process continues, zygote, through 8 stages, and eventually you have, think of a tennis ball that's hollow in the middle, and then eventually you have an opening. That opening is going to become part of the animal's systems. We know the prefix endo means inner, and the prefix ecto means outer. So you're going to have an endoderm found here in yellow, and the ectoderm found here in blue. Gastrulation, gastro, that prefix is stomach or gut, and we'll talk about more of that later. Ectoderm, shown here in red, is going to be the outermost layer. Endoderm is going to be in yellow, the innermost layer, and mesoderm is going to be the middle layer. This is giving you what these turn into. It's not important for your test. As you can see here on the far left, this is a flat organism, and we will talk about flatworms later on. This one does start to have a space in the middle, while this one does have a fully developed space. And what that space is called is a body cavity. You have body cavities, the area around your brain, which protects your brain as you walk and jump so it doesn't slam into the side of your skull. Many of you may have experienced a concussion, and that is what happens when you do have some brain damage. You have space around in your chest and in your stomach that allows for your stomach and your bladder to expand, as well as your lungs to expand when you breathe. If that space was not there, you would not be able to eat, and it would not be able to expand. Now, there are animals that do have all 3 tissue layers. Having 3 tissue layers is a word called triploblastic. Triplo, that tri prefix means 3. There's also a term called diploblastic, di meaning 2. The most simple of animals that we'll talk about are going to be diploblastic, or no tissues at all, and they cannot even be considered what we will talk about as an acoelomate. When we talk about an acoelomate, a pseudocoelomate, and eventually a true coelomate, these are animals that have all three layers of the ectoderm, the mesoderm, and the endoderm. This flat animal, or a flatworm, is considered an acoelomate, meaning it lacks a body cavity. You may recognize this prefix a from the word asymmetry, meaning no symmetry. So in this case, acoelomate means no coelom, or no body cavity. The phylum, which we will talk about later, is called Platyhelminthes, and those are our flatworms. These guys are allowing for eventually a development in organ system. Animals that do not have all 3 tissue layers cannot have organs because they do not have room for them. The next step in development is going to be animals that are considered pseudocoelomates. You may recognize the prefix pseudo from when we learned pseudopod or false feet. So in this case, pseudocoelomate means false coelom or false cavity. These are not quite developed. They do have some space. It's fluid filled, and it's going to be responsible for the transport of nutrients and oxygen, while these flatworms or acoelomates are going to rely on diffusion, but it is still not fully formed. So we have acoelomate, no coelom or no body cavity, and we have pseudocoelomates or false coelom or false cavity. What the false cavity allows for is a complete digestive system. As you can see here, this tube running through the middle allows for a complete passage from mouth to anus of food and waste. Acoelomates are going to have an entrance and an exit that happen at the same time, no complete path. The most fully developed and the most complex organisms, such as ourselves, are going to be true coelomates or coelomates. This means they do have a true fluid-filled body cavity. This protects and holds the organs in place and allows for expansion. Animals like Annelida, which will be earthworms and such, are the most advanced of all three phyla of worms. We also are going to be considered true coelomates. You can see here that this area is supported and allows for an opening to be consistent, so this tube, which is the digestive cavity, the tube that runs from mouth to anus, can be there, as well as other organs such in our bodies like our stomach, our intestines, our bladder, our liver, and also up in our nasal cavity and in our brain. In class, we did a demonstration and we took a large milkshake straw, we wrapped it in tissue paper, and then finally wrapped that in an index card. That represented the three tissue layers, the index card being a ectoderm, the tissue layer being a mesoderm, and the endoderm being represented by the straw. When you need a complete cavity, you want the same type of material to be on either side. So the way we represented a true coelom is we lined the tissue paper on the inside of the index card as well as the outside of the straw, creating a complete cavity encased in the same tissue. So to review, we had acoelomates that have no body cavity, pseudocoelomates that have a false body cavity, and coelomates that have a true body cavity. This all stemmed from the development of a single embryo of a single cell cleaving or multiplying into many cells, eventually forming what we know as an ectoderm and an endoderm. The final two terms that we learned were diploblastic, which means 2 tissue layers, and triploblastic as 3 tissue layers.